Bom, boa tarde a todos. Vamos dar início ao seminário dessa tarde, né? Eu tenho o prazer e a honra de apresentar a vocês o professor Fiorenzo Vetroni. O professor Fiorenzo Vetroni é da Universidade de Quebec, mais de um instituto, né? Instituto de pesquisa ligado à universidade. Professor Fiorenzo, ele já orientou. Ele foi contratado na Universidade de Quebec em 2010. E de 2010 até aqui ele já orientou mais de 80 pessoas, incluindo mestrado, doutorado e pós-doc, né? Então, só por esse número a gente percebe que o professor Fiorenzo é um professor extremamente ativo, né? Ele tem mais de 150 publicações em revistas de alto impacto. E quando eu falo alto impacto, é Nature, Science, nesse nível. Ele foi a primeira pessoa, o primeiro pesquisador a publicar um fenômeno que nós, que trabalhamos com lantanídeos, chamamos de upconversion, que é você joga uma energia menor sobre uma nanopartícula e obtém uma energia maior, né? Isso é chamado de upconversion. Não está se criando energia, mas ele foi o primeiro no mundo a observar isso em nanopartículas, tá? E o fenômeno foi observado pela primeira vez por um pesquisador francês chamado François Lozel, que infelizmente faleceu no início desse ano. Bom, o professor Fiorenzo recebeu diversos prêmios, inúmeros prêmios. Só para vocês terem uma ideia, né? Ele recebeu o prêmio da Natural Science and Engineering Research Council do Canadá, da International Union of Pure and Applied Chemistry, UPAC, da Royal Society of Chemistry, da ASM International. Ele recebeu prêmios na Itália, de prêmio chamado Gladiatore d'Oro. Recebeu o prêmio da Sociedade de Química do Canadá, da Sociedade de Eletroquímica, o prêmio Rutherford Memorial Medal em Química, da Sociedade do Canadá, de Química do Canadá. Ele é membro de diversas academias no mundo, né? Uma delas é o Global Young, College of New Scholars, Artists and Scientists of Royal Society of Canada. Em 2022, ano passado, né, ele foi agraciado como Fellow da Academia Canadense de Engenharia e Distinguished Fellow da International Engineering and Technology Institute. O professor Fiorenzo é um professor novo, né, mas ele tem muitas contribuições para o campo de nanopartículas envolvendo lantanídeos, né? Ele foi orientado pelo professor Capobianco, John Capobianco, também no Canadá, que é também uma referência para a gente no campo que a gente trabalha, né? E hoje o professor Fiorenzo tem colaborações com os principais pesquisadores do mundo na nossa área, Daniel Jaque, Xiaogang Liu, que é hoje um dos pioneiros, aí, um dos papas da área, né? Então é uma honra enorme receber aqui o professor Fiorenzo. Eu fiz a introdução em português, obviamente, mas ele vai falar inglês. So, Professor, it's a honor and a pleasure to have you here. Thank you so much for coming. And, well, I introduce you in Portuguese because so many data. Then I would like to make sure I didn't forget. Probably I forgot something, okay? But, well, thank you. The floor is yours.
So thank you very much, Fernando. Thank you for the wonderful hospitality during these few days. As I was mentioning, it's my first time not my first time in Brazil, but my first time in Sao Paulo State. So it's really a pleasure to be here, a pleasure to visit Fernando, and you know, I'll show you in a little bit where I come from. So this weather for me now is quite nice. So I'm very happy to be here. So as Fernando mentioned, I come from Montreal, Canada. So it's it's actually Montreal, and it's in French-speaking Canada, in the province of Quebec. Before I begin my presentation, I think maybe Fernando mentioned it a little bit. I want to first thank my collaborators, because everything I will show today is really a sort of international effort over many years, and it involves colleagues and friends from around the world, particularly a very old friend, Daniel Jaque from the University of Madrid, the group of Spaghini and Bettinelli in Verona, and other groups around the world. You see there's a group in Brazil here. Hopefully soon we'll put Unicamp as well. Singapore, pretty much everywhere, Europe, Asia, a very multinational effort. And of course, none of this would be possible without the people who have worked, who have come through my lab. Sorry. What did I do here? None of this would be possible without. There we go. None of this would be possible without the graduate students and the postdocs that have come through my lab. I mean, they're the real heroes in this story. I am privileged in that I get to fly to beautiful places around the world and give these seminars, but they're the people that did all the work. And all the best ideas come from them. So I come from Montreal. This is the city of Montreal. We have a park in the center of the city called Mount Royal Park, and it was designed by the same designer, by the same architect that designed Central Park in New York City. So basically, it's built around the mountain. So in French, Montréal means Mount Royal, or Royal Mount, and this is overlooking the city of Montreal. This is the skyline you see here. Actually, what you see here, this building here, this is part of McGill University, and that's part of their medical school. But this is the downtown core. It's actually changed in the last couple of years. There's some newer buildings, but this is the skyline. So this is in the summer, and as you can see here, this is in the fall, the same view. And the fall is very nice in Montreal, in Quebec. We have the changing of the leaves and all the colors. But you know, they say every good thing must come to an end. This is Montreal in the winter. And as you can see, it's, you know, romantic, I guess. If you're, for those of you who lived through the snow, the first snowfall is quite nice and very, beautiful, romantic. But then this happens. As you can see here, this is my car. And I was away, I believe, as you can see, the car here is nice and clean. Mine isn't. I was away during a snowstorm, and I came back a few days later and found my car buried on the street, so I had to dig it out of the snow. So, anyway. So I seem to be having a problem with the resolution of the screen. Let's see if this is better. Anyway, let's see if this is better. Okay. So I come from the University of Quebec. So we are in the Université du Québec. Basically, is a bit of a state school, if you want. It's a public institution with campuses around the province of Quebec. So the University of Quebec is sort of the umbrella that kind of oversees all the universities. And then we have universities that are spread out around the province of Quebec. Now, some years ago, the government of Quebec realized that if you didn't come from money, so if your family didn't have money, it was very difficult for you to access higher education. So the universities were in the big cities, so in the city of Montreal, in Quebec City, and in Sherbrooke. And so if you wanted to go to school, you had to move to those cities. And it wasn't easy for a lot of people, especially from people in farming communities. So they started this University of Quebec system, and they placed the universities in small towns around the province. And the big one is in Montreal. That's not where I am, but that's sort of the flagship university of the University of Quebec system. And then we have an engineering school, a polytechnic, we have a school for public administration, and a long-distance learning, a tele-learning university. But a few years later, about a decade later, they came to the conclusion, they realized that all these students that were in these schools here, if they wanted to access higher education, master's, PhD, well, they had the same problem. They had to go to the big city. And so they created my university, which is called l'Institut National de Recherche Scientifique. It's a bit of a strange name. For some reason, it doesn't have the word school, or in French, école, or university or université in it, rather institute. But we like to compare ourselves to the best, so we kind of compare ourselves to MIT, that doesn't have the name university in it, Massachusetts Institute of Technology. So we're kind of the same thing. So we are a research institute, university research institute. But the government of Quebec did something really well, and I typically don't give them credit often, but for this, actually I give them a lot of credit. What they decided to do was open up a school, a university, but that is not divided in departments like a conventional university like this one, or faculties. So there's no chemistry faculty, physics faculty, mathematics faculty. Rather, they decided to make centers. And each center is sort of dispersed around the province of Quebec. And every center is located in a sort of a location where the research is important to the region. So for example, we have a center for water, earth, and environment, which is in Quebec City, at the sort of the mouth of the Atlantic Ocean. And there they do all the hydrology work and environmental work and all that. So we have, over the years, the centers merged, and so we have now 4 centers left. Actually, most of them now are in or around Montreal, including mine. Mine is a center for energy, materials, and telecommunications. And in my center, we do everything that's related to materials, photonics, nanoscience, you name it. Of course, energy as well. And then we have we have another center that is that was started by a doctor named Armand Frappier, which is where all the virology, toxicology, everything biotechnology, everything biology related is currently being studied. So just very quickly, in my center, we have about 45 professors we offer various degrees, including, of course, the PhD. I made this mistake also before, but this is wrong, actually. This should be 100, I forgot a 5 here. We have about upwards of $150 million in infrastructure located on campus. So we're probably one of the universities that have the best infrastructure in the country, even though we're so small. We have a structure that kind of oversees all our equipment called the infrastructure of nanostructures and femtoscience. And that oversees sort of 3 or 4 large-scale centers. So we have the LMN, the Laboratory for Micro and Nanofabrication, which is basically your standard clean room. We're clean room from class 10,000 all the way to class 10. we just got a brand new E-beam, so we replaced our old E-beam. So we have basically a state-of-the-art E-beam that's just literally a couple of months old. We have another big center here called the Advanced Laser Light Source. The Advanced Laser Light Source is a national facility. It operates a little bit like the synchrotron that I visited earlier this morning, where in order to get beam time, you have to write a proposal. There's an independent body committee that analyzes it and then awards you beam time. And basically you have access to the femtosecond regime, to the femtosecond rainbow, femtosecond pulses from, various, from the X-ray all the way to the terahertz. This is actually, we received a large grant to update these lasers now. Actually, no, yeah, these are the right ones, ytterbium YAG. They used to be titanium sapphires, now they're ytterbium YAG lasers. And we also have an RPs, which is only one of two in Canada, and the most powerful laser in Canada at 750 terawatts. Now we're working to build that to 1 petawatt. We also, the only basic institute in Canada that has dynamic TEMs, so we have two of these dynamic and ultra-fast TEMs here, which basically give you not only spatial resolution, but also temporal resolution, so you can see how a material changes in real time. And then we have a bunch of other equipment around the labs. Okay, so with that said, let me talk a little bit about what I do. So I work basically kind of in the field of theranostics, and that's we do other things as well, but that's where I kind of enjoy the most. And basically what we're trying to do, so the overarching, the theme of the research is to build these multifunctional nanoplatforms where we can combine two or more of these modalities onto a single platform that is essentially triggered by light and triggered by near-infrared light. And almost always, there's a nanoparticle at the core of this platform that is based on rare earth ions or lanthanide ions. So with the exception of magnetic resonance imaging, all these modalities here are light driven, okay? So we want to combine these together and see, and see if we can generate new properties, new findings. So for those of you who don't know, theranostics basically is the combination of therapy and diagnostics onto the same platform. So if you have some sort of illness, like a malignancy, you use some sort of stimulus. In our case, it's always light, mostly light, yes, almost mostly light, right? And so you can detect this malignancy, and on the same platform, then you can treat it and eradicate it, and hopefully the patient will one day recover. So there are many types of nanoparticles that are being used for this. Many of you are familiar with these, from organics to carbon materials, of course, the quantum dots, metals. But so all these nanoparticles, have been used and have been reported, again, for these types of applications in theranostics. But in my opinion, and hopefully I can convince some of you, I guess many of you already work in this field, so you don't need convincing, but hopefully I can convince the rest that the rare earth ions, or so the rare earth doped nanoparticles, are really, my opinion, probably the most versatile and the best for these types of applications because of their optical properties. So I don't need to show this here in a chemistry institute. We all know the rare earths. What you may not know, though, is this here. This is called the Dika diagram. For us rare earth spectroscopists, it's a little bit like the Bible, and we always refer back to this diagram here. What you see on the x-axis, these are the tri-positive, so the 3+ rare earths, rare earth ions, energy in inverse centimeters. And what you see, all these lines here, these are the 4F electronic energy states. And you see that there are very many states, very close together. So if you take an ion like, for example, erbium, let me go to the next slide here. If you take an ion like erbium, you see it as a ground state here, which would be here. There's an intermediate state about there, which would be this guy. And then if you go up this ladder, there's another excited state about there, which will be this guy here. So the energy between the ground state and the intermediate state is pretty much equal to the energy between this intermediate state and this upper state. which means that you can use a femtosecond, which means you can use a near-infrared laser to populate this upper state, right? So you have one near-infrared photon come in, it populates you, it basically excites through ground state absorption the ion from the ground state to this state. Because the lifetime of this state is long, typically milliseconds, hundreds of microseconds, there's a very high probability that a second incoming photon will propel you to that upper state. And then you emit a higher energy photon that's essentially equal energy to these two photons. It's not always the case. It's a very, very simplified version of what we call up conversion, very simplified, but that's the case. So this is a two-photon process. We've demonstrated in the past up to 6-photon process. Now, so that's not it. That's not all there is. So when the ion is excited to this intermediate state here, of course, as we said, it can go up, right? But the ion can non-radiatively decay to this lower state here and then emit a lower energy near-infrared photon, which we call downshifting. Okay? So with a single excitation beam, a single excitation source, you can have both visible, ultraviolet visible upconversion, and then you can have near-infrared downshifting emission at the same time. Okay? And this is important, and hopefully you'll see why in the coming slides. Now, I should mention that this is a type of multiphoton excitation, very different from conventional multiphoton excitation that many of you may be familiar with in organic dyes or in quantum dots. In this case, these are real excited states, so it's a sequential absorption of photons, so they go step by step, and so you can do it with a very inexpensive near-infrared laser. In other types of materials, there is no real excited state. There's no real intermediate state. So you need an ultra-fast laser, like a femtosecond laser, and the ions and the photons have to arrive almost simultaneously in order to propel you to that upper state. Okay? So very different photophysics. This is much more efficient. So we can get all the colors of the rainbow. We do it differently than the quantum dot people. We change the doping, we change the dopants. We change the, we co-dope. We change the concentration of dopants, and so we can pretty much get all the colors up until, up to white light, just by playing with these, just by playing with this concentration. And so I think I also want to mention that we work principally with fluorides, okay? And I need to mention here that the rare earth ions are dopants, okay? So the nanoparticles are not based only on rare earths. The rare earths are kind of impurities that we intentionally place inside these crystals, and the crystals replace, the rare earths replace the metal stoichiometrically inside the crystal, okay? Now, why fluorides? Again, if we go back to the Deca diagram, I point out to the plethora of these excited states, of these 4F electronic energy states, which means that the upconversion, for example, in most of the luminescence is very sensitive to the lattice vibration, to the phonon energy of the material. If the phonon energy is too high, what happens is once the ion is excited to the intermediate state, when you have high phonon energies, the ion will non-radiatively decay down to a lower state. Now, if you look at upconversion, you see that there is no state above with a similar energy gap. And so that significantly decreases the intensity of the upconversion, okay? And all the other luminescence as well. So we need materials that have low phonon energies, and at the moment the fluorides are pretty much some of the state of the art. And we worked with most of these in one form or another over the years. So to sum up, the rare earth nanoparticles are pretty versatile in terms of their optical properties. As I showed you, we can do upconversion, we can do downshifted near-infrared emission. They're photostable. So I think Fernando told me that these were changed to LEDs, but in the past, in fluorescent bulbs, you had rare earth ions in there. There are rare earth ions in lasers, and as you saw earlier, ytterbium YAG, neodymium YAG. They're used ubiquitously. They're used everywhere, so they're very, very photostable, okay? So you can pump these materials for months and years without any noticeable loss in signal intensity. And of course, for applications that I'm interested in on the biology side, I'll say they're biocompatible. Now, there's still a lot of work to be done in terms of biocompatibility. Still a lot of work to be done to determine how a nanoparticle interacts in the body, how it's excreted from the body, and so forth. But from the data we have today, in terms of cytotoxicity and even in vivo toxicity, I can confidently say that we can use them in a living specimen at the moment. Of course, anything can change once we get new data. And I know Fernando is working on something along those lines, so I'm really looking forward to those results. So, as I mentioned, we get upconversion and downshift luminescence. So, what could you do with these nanoparticles? Well, we've done a bunch of work on deep tissue imaging, and I'll show you some stuff. We've done work on sensing. We've done work on sensing particular bioassays, and a lot of work on thermometry. This is one of the big topics that my group has tackled over the last 10 years, maybe, on thermometry, and I'll show you some examples of this. And a lot of work also on using light to trigger drug release, photodynamic therapy, and I'll show some results in the coming slides. So my adventure in this field started 23 years ago. Actually, this paper was published in 2000, but it was a few years before it started. So I would say about 25 years ago. And here, this is where we first showed upconversion at the nanoscale. And I spent my PhD, I could pretty much sum up 6 years of work here. This was my PhD. I didn't even need to write a thesis. I could have just showed this and explained this, and that would have been my defense. So I studied all the mechanisms that led to upconversion at the nanoscale. Spent 6 years in the dark in a laser lab with no windows. So to all you students in the room, don't give up. It gets better, I promise. So that first paper was, if I can just go back here, that first paper was on sesquioxides, on yttrium oxide. And at some point during my PhD, we got the idea that these materials could potentially be used in biological applications because we're exciting them in near-infrared light, and near-infrared light has tissue penetration capabilities, as I'll show. So maybe, there's some potential there in biology and biosensors. But the problem is that the materials we were working with were made with a very crude combustion process. So basically, you mix the oxides in glycerin, and then you heat it, and it explodes, and you get these nanocrystallites, which was cool for fundamental studies, but for practical application, wasn't cool. And of course, we wanted to move to fluorides because fluorides, as I mentioned, the low phonon energies and so forth. So after a few years, we published this paper here where we published the synthesis of these materials. And I want to point out this here. And you see that so these were the first nanoparticles that we reported some years ago. And you can see they're not very nice. But, like all good things, you have to keep at it. And again, to the students in the room, don't give up, keep at it. Over the years, we've automated our synthesis where we have, you don't see the syringe here, but we have a syringe pump with a syringe and a needle, and then we basically heat our precursors, we drop them in there, and we can control pretty much all the properties you need to control. Injection rate, temperature here, cooking time here, spin rate. We can control everything, and I'll show you, I think, some examples of what we can do. I won't get into the chemistry too much, but this is a thermal decomposition process where we decompose trifluoroacetate precursors in the presence of a coordinating ligand, oleic acid or oleamine, and a non-coordinating ligand like octadecene. We heat that up, and then we decompose the trifluoroacetates. This is what they look like in real life. You can see the near-infrared, you can't see the near-infrared laser, that's why we put the card, so it's a 980 nanometer laser. These are nanoparticles in water. We have the blue emission from thulium ions, that green emission from erbium, and then we can mix and match to get other colors, depending, again, as I mentioned, depending on the concentration of the dopant, the co-dopant, and so forth. So we can get all the colors we want. And just as an example of the type of nanoparticles we can make, well, again, we have very strict control over the morphology and over everything, size of the nanoparticles, so we can make, hexagonal particles, we can make spherical particles, we can even make short stubby nanorods. We've made longer nanorods after that. But it's important to point out that they're all very crystalline, okay? But not only are they crystalline, it's also important to notice that the particle size is very uniform. And for applications in biology, especially in vivo, this is critical, because different size nanoparticles will behave differently in the body. And so, when you're working with living specimens, there's so many variables, and if you have different size, if you have like a wide particle size distributions, you have big particles that will clear from the body using one mechanism, or go into the cells using one mechanism, and then cleared smaller particles will clear differently or use another mechanism to get into the cells, so it becomes a mess. So when working in vivo, just as a suggestion, you want to make sure you have a very tight particle size distribution. So, as I mentioned, we work with theranostic materials. And so one of the challenges we have is how do we get our nanoparticles to the site? I'll show an example later on of the latest stuff we're doing based on that. But we have a collaborator in Singapore who makes these really cool capsules. So they're silica capsules, but hollow. And we can incorporate whatever we want inside, and we can make them as big as we want. And so one of the things I was really interested in doing is incorporating one upconverting nanoparticle and a photosensitive dye, zinc phthalocyanine. Okay, so zinc phthalocyanine is, as I mentioned, a photosensitive dye, and it converts light. It converts light to reactive oxygen, and reactive oxygen is toxic to cells. And so this is something in cancer therapy called photodynamic therapy. The problem is that this zinc phthalocyanide, this ZNPC here, which is used in the clinic already, only absorbs visible light, and so which means that it can only be used on illnesses or malignancies that are on the surface, right? Because visible light does not penetrate your skin. So, of course, what we want to do is use our nanoparticles that can convert near-infrared light that penetrates the skin to higher energies, and use them as sort of a transducer, as an intermediate, if you want. So we excite the nanoparticle, it generates the visible light inside the body, and then that light is absorbed by the photosensitizer, which then generates the reactive oxygen. So all that to say, this is I guess, the important data is right here. So this is the actual structure we made. You can't really see it, but anyway, this is the structure right here. And this is the important data here. This is where this is capsase. So this is, the cells are stained with a dye that detects cell apoptosis, so when a cell dies. And you can see that when we have our nanoparticles with the dye in the nanocapsule, we get pretty much the cells die. And that was important for us, and that was an important finding. So if we just change topics for a second, one of the things that is a big interest to my group is thermometry, nanothermometry. Why? Many years ago, I was visiting my friend Daniel Hake in Madrid. And we were talking, we were, I think, having supper, drinking, and at some point the discussion turned to science. And after some discussion, we realized that the luminescence of the rare earth ions has been known for a century. And we also know from probably 100 years ago that some of the emission bands are sensitive to temperature. So could we use these nanoparticles as non-contact, sort of remote, minimally invasive, minimally contact or remote control, whatever you want to call them, thermometers? But why? Why do we care about that? I think there's two main reasons. One is a little bit more fundamental, and one is more applied. Fundamentally speaking, everything in nature is controlled by temperature. If you look at cell denaturation, cell differentiation, you name it, is controlled by temperature. So if we can make temperature probes that we can send in and monitor temperature remotely, then maybe we can study new fundamental biological processes. The other one is more applied. We know that the first sign of most illnesses, whether it's cardiac, whether it's cancer, whether it's infection, you name it. The first sign of most of these illnesses is a thermal gradient, is a rise in temperature, a local rise in temperature. So if we can design, again, sensitive thermometers, then we can potentially use it as a detection tool to detect disease. And that falls into sort of the general scheme of my research group of theranostics, the diagnostics part. Okay. Now, of course, there's a whole other community. I think some of you here work with plasmonic materials. There's a whole other community out there that work with what we call nanoheaters, plasmonic materials that can convert light to heat. And people are using them to kill cancer cells, photothermal therapy. And so, you know, the issue is we've done work with that. I'm not going to show it in this presentation, but I can show it if any of you have questions. But we've done work along those lines. And if you have like a really good, like a gold nanorod that has a very high heat conversion efficiency, you risk not only it's so good at heating that when you excite the nanoparticles, you risk killing the healthy cells around, and then you start to get side effects that you don't want. So we thought, how about using thermometry in conjunction with photothermal therapy, where we can both monitor, where you can heat, and then in real time monitor temperature. So that's kind of the preamble of why we care, why we were so excited about thermometry, 10 years ago. So there's various types of optical thermometers. We really focus on the luminescence type, the fluorescence nanothermometers, luminescence nanothermometers, and even within that category, there's various subcategories. We have done a lot of work and continuing to do work on band-shaped thermometry. In this particular case, as I'll show, you have two bands that change as a function of temperature, and we can correlate that change to temperature. And very recently, we started working on lifetime thermometers. And I believe I will show an example. So how do the band-shaped thermometers work? Well, the classic ones are erbium, ytterbium. So this was a figure out of my thesis from long ago when we were working again with the oxides. In this case, it was lutetium oxide. We weren't doing thermometry back then, but we were just studying, again, the fundamental mechanisms of how we populate these excited states. And so if you look at these two emissions here, so this emission is from 2H11 halves. This big guy here is this emission from 4S3 halves. So what happens is you have this upconversion process, or it can occur also if you excite at a higher energy, but regardless, you basically populate the state here. It will non-radiatively decay to the 2H state, right, through multiphonon decay. Now, because these two states are really close in energy, this state will immediately non-radiatively decay to the 4S state, and we get this emission here. So it's this big guy here, okay? But because these two states are essentially, they're so close in energy that they're in a thermal equilibrium, okay? So basically, they obey the Boltzmann law. So they're in thermal equilibrium, so the lower state populates, can repopulate the upper state, right? And then you get this emission here, right? And this repopulation, again, it's Boltzmann, so basically it's controlled by temperature. And so what happens is when you decrease the temperature, as you see here, the rate of rethermalization or thermalization, essentially, the probability of this happening decreases until you pretty much kill this emission, right? And so now you have this peak here, and so obviously these ratios change. Of course, the opposite is true. If you heat, then this becomes much more probable, so you're feeding the state at a greater rate, and this increases at the expense of this guy, right? So we can correlate that ratio, so what we call ratiometric nanothermometry, we can correlate that to temperature. And so with Daniel Haki a few years ago, we did just that. We looked at living cells, and we're really the first to show that we can determine temperature in living cancer cells, of course, in the physiological range, based on the ratio of these two peaks, with relative accuracy. Sub-degree resolution for sure, and we've since pushed the envelope even better. So this is ratiometric. The problem with ratiometric, it's really easy to do. So it's accessible to everybody because all you need is a spectrometer. But when you get in the body, there are other factors that will affect your ratio. And those factors that affect the ratio can throw off your calibration. So talking with one of my colleagues who has the office next to mine, who's an optical engineer, I told him the problem, and I told him that lifetime will be less affected by the surroundings in the body, right? And he said, okay, can you do FLIM? I said, well, we can do FLIM, which is fluorescent lifetime imaging microscopy. But, you know, it's a big system, very expensive, $1 million instrument. We don't have one. And plus you have to heat, you need a near-infrared laser that may not be accessible. He said, okay, I'll build you one. So he built me this, what we call the split system. It's been patented. Now, I don't understand too much the optics, but essentially, there's probably those of you in the room that know more about the optics than me, but basically it uses a very inexpensive continuous wave laser. We chop the light in order to be able to get the lifetime, and then he does something with masks and that I don't understand. But for me, what was the important thing was, can we use it? Can we really use it to detect lifetimes as a function of temperature? So we made some various nanoparticles. Different morphologies, different architecture, core shell, 2 shells, 3 shells. And the more we shell, the more we protect the core, and the more the lifetime changes, the more the lifetime gets longer. And so this split microscope, which is fantastic because it's single shot, wide field, as you can see here. This is 250 micrometers. If you look at the movie here, these are the nanoparticles. These are different nanoparticles with different lifetimes. CAN for Canada. If you look at, I don't know if this movie, I guess it was too fast for me. Do you see the movie there? Yeah. So as you can see, in real time, we see the decay of the nanoparticles in a wide field. This was our room temperature, and then as a function of temperature, again, I missed the movie, but sorry. You can see the movie, I hope. So that's our room temperature, and you see here, this is the green emission, the red emission, as a function of temperature. And so we can use this to actually image in real time. The lifetime in real time. These are masks, of course, but it works so well. This has been patented. Now we've built another system based on this one that is for animals. So we got when we showed it to the cancer, the Canadian Cancer Society people, they were really excited about it. Particularly in the domain of melanoma. Melanoma is very hard to detect because melanoma, skin cancer, the detection relies mostly, the early detection relies mostly on the oncologist or the dermatologist that's looking at the moles on your skin. They have like a technique to do it, but a lot of it is visual. So they were really excited if we can use this in order to be able to detect melanoma, because this is through upconversion, remember. So we're exciting in the near infrared and we're looking at the emission in the visible. So they were, which is ideal for skin malignancies. And of course, we also looked at cells, and you can see we can actually track the cells as they're moving in real time and different lifetimes where we have particles and where we don't have particles. So a very powerful system that we're really, really happy with. And I'll talk about an even newer system in the coming slides. So everything I've showed was at upconversion, where we excite in the near infrared and we get visible light coming out. But let's forget about upconversion for a second. Remember I told you that the rare earth ions also have near-infrared downshifted emission, so near-infrared luminescence. So, and this is important because the skin has several windows that are optically transparent. So there are 3 windows in tissue where light can penetrate, okay? And the ideal ion for this is neodymium. The neodymium ion, because I think my batteries are dying on my pointer, but you can excite it at 800 in the first biological window, right? And then it has various emissions in the second biological window, right? So you excite in the first, and then you have emissions in the second biological window. So you can use that potentially for imaging. You can use that for thermometry. 800 is a heating-free excitation. I forgot to mention that we use 980 typically for upconversion. 980 heats, okay? It's absorbed by water. So we need to take that into consideration when we're doing our experiments. We don't have that problem with 800, or at least it's much, much less drastic. So we consider it a heating-free wavelength. And of course, neodymium has emission in the 2 biological windows. And can we use it for imaging and thermometry? That's the key question. So, as ever, we were interested in seeing if we could. Again, always interested in a theranostic platform, a multifunctional platform where we can combine, imaging, not imaging, but diagnostics and therapy on a single platform. Again, we took advantage of those really cool nanocapsules that my colleague in Singapore makes, but this time we added downshifting nanoparticles, so neodymium-based nanoparticles that will emit in the near-infrared. We excite them at 800, and they emit in the near-infrared. And we added this dye here, base-substituted dicyclohexylamino-pyrroline diamide. This is a collaboration with a group in China where they're working with this dye here for photothermal therapy. So again, we wanted to see if we can control temperature, if we can monitor temperature as we heat the dye. So as you see here, so we heat, we excite with near-infrared, we heat, and then we have temperature sensing, and of course we have imaging. I haven't talked about the X-ray stuff, but the nanoparticle contains lutetium, so we also do X-ray imaging and SPECT, I believe it is. But anyway, we'll just focus on the optical properties now. So these are the nanoparticles inside this very thin, you can barely see it, but they're inside the capsule and with the dye, and you can see that we can get heating. This is laser heating, so this is a laser heating of the nanoparticle with the lutetium. Again, we have this temperature change, and we can control temperature in real time. We can monitor temperature in real time. Of course, the important part is here. We've stained, again, the cells with capsaicin, and you see that when we have the dye and we're heating, then obviously they fluoresce because the dye fluoresces, and so we've induced apoptosis, so the cells die. So we can actually heat and monitor in real time. But before that, we looked at imaging. We looked at imaging using neodymium, and we wanted to see if we can do whole animal imaging with neodymium-based nanoparticles. And we did, of course. What we didn't know is that we got this really broad absorption in our spectrum. And so when we actually did the imaging of the whole animal, we got this blob here that incorporated the background. And we didn't know why. It took us a while to figure out that the food that the animal eats is actually fluorescent. So of course we had to, in later studies we had to feed the animal different foods, but in this particular study we just shifted the acquisition wavelength beyond 1100 here, and we start to see some fine structure in our animal. So basically, we showed that we can use the near-infrared in a theranostic platform. We showed that we can use it for imaging. Can we use it for thermometry? Yes. So again, neodymium. I didn't mention that the thermometry with neodymium occurs in a slightly different manner. It's still ratiometric, but rather between 2 excited states. It's within the same excited state, but they're with their Stark states. So the excited states split further, and these, what we call Stark states, are actually Boltzmann coupled. So we can actually, again, calibrate and then determine temperature. So this is in the first biological window, and I'll show you the results. But we've also then since worked in the second biological window, we've done thermometry, and in the third biological window as well. So we've covered all the windows when it comes to thermometry. But just to show that if it worked, this is a tungsten wire on a surface. We run a current through it. Of course, our nanoparticles are spin coated here. When we run the current, and we see where the hot spot is, of course, is where the current is passing. We've looked at microfluidic channels. In this particular case, we're using 1500 to heat a channel containing our nanoparticles. And when we heat, the water is absorbed. And then we use 800 to map. And of course, we see the hot spot is at the focal point. So that works. And then in the experiment that kind of interested me the most was in a tissue. We took a chicken breast tissue, we heated it, and we looked at the dynamics of the heating and cooling and the thermal map, and it worked. So we're good. So that's all ratiometric. My colleague likes acronyms. So speaking with my colleague again, after we published that first paper, and we said, okay, where do we go now? Well, if we want to do biology, I told him we need to push the instrument to the near infrared. So can we design a similar instrument that's in the near infrared? So obviously he did. He's brilliant, I have to say. When it comes to optical engineering, he could build anything. And so he built me this system here. This is essentially a big streak camera. Same concept as in the split, although he named it a different acronym for whatever reason. But so this is all near infrared. So it's an interchangeable laser here, 980 and 800, that goes through some various optics and gets chopped. In this case, we're using a modulator. We're not using a chopper, but we're pulsing this, and it goes through a bunch of optics into our near infrared camera. So this is the pride and joy of the device, this near infrared camera here. So this detects lifetimes in the near infrared. Again, wide field, ultra fast. We did the same experiment. We designed a bunch of different nanoparticles with different hosts, different dopants, with different lifetimes. We wanted to see if we could map, again, in real time, large scale. And you see here, so this was a really cool experiment, which we're kind of exploiting for anti-counterfeiting measures. In this particular example, my student combined a bunch of different nanoparticles with different lifetimes. And as you can see, it can map all the different lifetimes. So this is a map of all the different lifetimes of all the different nanoparticles that are on this star mask here, which was super exciting. And of course, we can do the biological work. In this particular case, we looked at beef tissue, and we were able to image. I don't show it, but we were able to image wide field the thing. So this has recently been accepted in advanced science. This is actually the first generation. The second generation has a grating, and now we're able to do spectroscopy also. That's the next step. Okay, so that was near infrared. Let's come back to upconversion for a second. So I just want to remind you that with a near infrared beam, we get upconverted emission, and we can also get downshifted emission. Sorry about that. My pointer is going a little bit crazy. Downshifted emission, right? So again, it speaks to the versatility of these materials. Near infrared excitation, two types of emission. So some years ago, we borrowed a page from the quantum dot community. Okay. I'm very interested, I was telling Fernando today, I'm interested in multi-architecturing. I'm very interested in, when you have a nanoparticle, when you have various shells, and every shell has a different function, I find that really cool. And by changing the shell architecture, we can get new properties. So some years ago, we did an experiment where we did a core shell, and whatever, it worked. We got really bright emission. The idea was to improve the emission of the upconversion. But that got me thinking. That got me thinking about multi-architecturing, how we can use the shell to our advantage. So one of my postdocs at the time, Marta Quintanilla, who's now a professor in Spain, she made this nanoparticle here, which is basically you have thulium, which if you recall from my little movie there, upconverts to blue, so near infrared to blue, erbium, which upconverts near infrared to green, and then we passivated the surface in order to improve the intensity. So with that said, this is the spectrum, okay? And you see where the most intense peaks are from the thulium ion that's in the core. And it makes sense because the core is protected by two shells. Now, she made the exact same nanoparticles, except she put erbium here and she put thulium there. So the exact same nanoparticles with the exact same dopant concentrations, except the only thing that changed was the position of these two ions. And the spectrum is completely different, right? Where now the most intense peaks come from erbium. So, you know, if you think about it, it makes sense, of course, because the core ions are protected by two shells. But these are the same nanoparticles. They were made at the same time. They have the same rare earth dopant concentration, same size, same ligands, same everything. The only thing that changes is where we put these ions in the architecture. So I want you to keep that in mind for the coming slides. So this core shell architecturing is, I think, something that's very powerful in this field. Some years ago, we showed that with these lithium-based particles, which have become now the workhorse of my group, we can, using 980, we can get like a really beautiful spectrum where this is a 2-photon emission, 3 photons, 4 photons, 5 photons, and this little guy here is 6 photons. So this is inconsequential, of course, it's a little blip, but it's 6-photon emission, and that was at the nanoscale, so that was pretty exciting for us at the time. But what really excited us was this peak here, this very strong UV peak, which we realized that we can use it to trigger another light-activated modality. Most of the photochemical processes or chromic processes are triggered or excited by UV light. So we thought, okay, can we exploit this? Can we harness this? So we made these nanoparticles again, and now with a colleague at McGill University who's a hydrogel expert, we did some chemistry, we grew silica, and then we anchored a chirosan-based hydrogel. But the hydrogel was modified with this nitrobenzyl group here. And this nitrobenzyl group will essentially undergo dissociation when it absorbs UV light, okay? And so in the shell, we put a cargo, right? So what we were going to deliver. In this case, it was just a model macromolecule, bovine serum albumin, which we tagged with a fluorescent dye. So the moment we turn on the laser, well, actually, you see that the UV light, the UV emission here decreases, and it decreases as a function of the nitrobenzyl concentration because it's being absorbed by this photolinker. Okay, so the more we added that, the more we quench the UV, which tells us that, of course, it's being absorbed. And so the moment we turn on the laser, we have release. So what happens is the nitrobenzyl group absorbs the UV light, it dissociates, the bond breaks, and essentially the hydrogel in the vicinity collapses, and the cargo is released. So it's really cool. It was really controlled, because the moment we turn off the laser, we got no release. But there was some problems with it. There's a couple problems with it. First problem is that we were using this is 800, this emission here is in the first biological window, so we were using that for thermometry and for imaging. Why is that a problem? Because we're using a single excitation wavelength, we're using 980. So if we wanted to image the nanoparticle right after injection, if we wanted to track it as it went in the body before it got to the site of interest, the moment we turn on the laser, we also generate the UV emission, which then triggers the release. So that was a problem. If we want to, for example, after you induce a therapeutic process, you release the drug at the site of interest, then you want to image it, let's say the next day, or 2 hours later, whatever it is. Again, the moment you turn on the laser to image, well, then you're releasing more drug. So it's an optical problem. Could we solve that? The other problem is that the particles are really big. In order to get this large UV emission, we needed almost 100 nanometer particles. So I'm going to been talking for a lot, so I'm going to try and go a little faster. So remember this decoupling process, this architecturing process. So what we decided to do is to see if we can decouple using this core multi-shell engineering, if you want, if we can decouple the therapy, the drug release, or photodynamic therapy, or the diagnostic part, nanothermometry or imaging. And so we did. I had a really bright PhD student who was now a postdoc at Berkeley, Artyom, and this was Artyom's work. And so Artyom devised this nanoparticle here, which is basically kind of modeled in this cartoon here. And if you look carefully, you may not see it, but there's a bunch of people around an operating table with a patient there, and you see kind of the laser going in from here, right? And then you have a bunch of people here on a kind of a table with a microscope, and they're kind of looking into the microscope. So this is the therapy, right? They're fixing the patient, and here they're trying to discover what's wrong with him. The laser's going in here. But between the people doing the detection and the people doing the therapy, there's a brick wall, and that's important. We built that nanoparticle that looks like this. So essentially we have an upconversion core, we have an undoped shell of a certain thickness, and then we have neodymium on the outer shell. Okay? So we've basically, everything I've talked about, we put together. Upconversion in the core to trigger, neodymium in the shell to image and for thermometry. Separated by a wall. The rare earth ions are really, really good at talking to each other, right? So if we can keep them spatially segregated, then they won't talk to each other. And this is what it looks like in real life. When we excite at 980, we have the UV emission that we need for triggering, for our therapeutic process, and nothing in the near infrared. When we excite at what we call the diagnostic wavelength, 800, then we have no upconversion, so we're not triggering any release, and then we have the neodymium peaks for imaging and thermometry. So very quickly, we combine these nanoparticles with another photosensitive dye, CE6. CE6, same concept. When the important data is here. When we're at the what we call the therapeutic wavelength, so when we have our nanoparticles with the dye, and we're exciting at 980, and we see these red dots here, this is indicative of apoptosis, so the cells are essentially dead or dying. So that's really good. With two different cell lines. You don't actually see the red in this one, but take my word for it, there's a lot of red here. This is in a 2D cell culture. If we look at 3D cell culture, same thing. When we're at the therapeutic wavelength, we see the red, which is indicative of cell apoptosis. So the cells are dying. And of course, when we're exciting at 800, the cells are perfectly happy, no release of any drug or any reactive oxygen, and off they go. So I'm going to skip this because I've been talking for long. We did the imaging and we did the thermometry part, so we have a real theranostic platform on our hands here. Same concept, decoupled next generation. I'm going to skip this. So the problem, we tackled the optical problem of decoupling, but we didn't. We didn't tackle the problem of size. So we wanted nanoparticles now that were really, really small. So I had a postdoc from Singapore, who spent almost 2 years on this problem. Really go back to the basics of the synthesis, that we developed many years ago. Can we make these lithium particles small and bright? And so after 2 years of work, these are the gold standards, so they're about 100 nanometers. And so, it took a long time. And what we figured out was, if we do a multi-architecturing, if we put, this should be lithium ytterbium fluoride, but if we do a multi-architecturing, we can get over the problem of this non-radiative decay. So what she did is she made these lithium ytterbium particles. Now, this is counterintuitive. For those of you that work in the rare earth field, this is counterintuitive because typically when you add too much rare earth, too much ytterbium ions in the matrix or as a dopant, what happens is the ytterbium ions talk to each other. They energy transfer among themselves, and that leads to quenching, self-absorption. But what we showed that if we put ytterbium, we're absorbing more near-infrared ions. But because we're shelling with lithium yttrium fluoride, the effect of the shelling and the absorption of the near-infrared ions is greater than the losses due to self-absorption. Okay? So what happens, the end result is that we have small particles, sub 20 nanometers, okay, as you see here, that have ultraviolet intensities that are higher, much higher, than the gold standard of 100 nanometers. So that was super exciting for us, and this has become really now this material here is probably the material we work on the most in my group. And we're using it, so these lithium ytterbium, we're using it in a new study where we're trying to target tumor cells. And this, I'm going to end with this. This is a big problem in the field. For those of you in the field that are in the sort of nanomedicine type field, the biggest problem is getting your particles where you want them. The body is really, really good at detecting a foreign substance, isolating it, and keeping it apart. Okay? So we want to figure out a way to evade the body's immune system. COVID, for example, perfect. Does that perfectly. Okay? So one of the things we thought of, can we actually use stem cells? Can we actually use something from the body to actually deliver the particles? Stem cells are great for this because stem cells will home in to a tumor. Okay? If you have a tumor and you have stem cells in the vicinity, the stem cells will go straight to the tumor. Don't ask me why, I'm not a biologist. This is what my biologist colleagues told me. So we use two types of stem cells, ethically sourced. This was important for me. We wanted stem cells that were obtained from an ethical source, so not from fetal tissue or anything, that were obtained both in surgeries. This is from plastic surgery, and this is from I forgot the word, but anyway, from bone marrow surgery. Okay, and so again, we thought about doing the same thing, incorporating but using the stem cells as our delivery vehicle. So we used again our decoupled nanoparticles, slightly different configuration. I won't get into the details again in the interest of time, but same concept. Okay, decoupling, two different wavelengths, no therapy. Okay. And the result is this. The result is when we incorporate the stem cells, the nanoparticles in the stem cells, and then we put the stem cells in a matrix, in a cancerous matrix, they will hone in directly to the cancer, as we said. And so what we again, the next challenge was, now how do we get these nanoparticles out of the stem cells and into the cancer? So we thought about doing a two-step excitation process. So the nanoparticle, the stem cells went to the tumor site, and then we irradiated the tumor site. And by the irradiation, now we're starting to give off reactive oxygen. But reactive oxygen is also toxic to the stem cells, right? So it kills the stem cells. So we wait a day, so we do a first irradiation and a second irradiation. Then once the stem cell dies, we irradiate the tumor directly because now the particles are directly into the tumor, right? And again, we got really good results where we're inducing cell apoptosis and cancer eradication. So now the next step is to do this in vivo. We of course worked in 3D cell cultures, same thing. When we mix the stem cells in a 3D matrix, same thing. It works really, really well. Okay, with that, I'm done. I hope I've convinced you that rare earth doped nanoparticles are really versatile materials. We can use them for a bunch of applications. I've showed you, I've only scratched the surface of what you can do with these materials in terms of drug release, in terms of imaging, in terms of sensing, in terms of thermometry. There are also other applications that we've kind of tackled in environment, in energy, which the luminescence properties of these materials are really good for. And I really believe, I just want to end with this, I really believe that these types of nanoparticles could really form the nucleus of these multimodal theranostic platforms. So with that, I thank the funding agencies really that paid for this work, that gave me the money in order to do this work, from provincial agencies, national agencies, foundations, international agencies, cancer societies. Without them, it wouldn't be possible. I thank you sincerely for your attention. I apologize if I spoke for too long. I'm happy to answer any questions. If you're interested, these are my coordinates. We're always looking for good students, good postdocs, so welcome. Thank you.
Thank you, Fiorenzo, for this sparring. This sparring talk. So we hope you have questions for Professor Fiorenzo. Se vocês tiverem questão em inglês, também pode fazer. A gente traduz aqui para o Fiorenzo. Tá, então, abro para as questões. Yeah. Ayrton.
Thank you for the presentation. Thank you. I'm just curious, like in the Tullian emission that you showed with the 6 photons absorption, if you could detect that emission in all the power densities that you used, or it depends on the power density?
Yeah. So it's a multi-photon process, very high order process. So you need a very high power density to see that emission, of course. We also showed, like you guys, at some point we can sensitize gadolinium. So that's pretty cool. I think that's something really cool. That you can sensitize gadolinium at the nanoscale, fantastic.
Any more questions? René, here.
Thank you for your presentation. It's more of a fundamental question, actually. It's just that I didn't quite understood how come this upturn phenomenon, how come it generates more energy than it receives, really? If it's not a too loaded question.
No, so you're asking how we generate UV light from near-infrared light? Yeah, okay. So basically, if you look at this thing here, okay, it's a multi-photon process. So it's the addition of photons. It's a sequential addition of photons. Because this excited state here, or any of these excited states, their lifetime is very long, okay? So when you excite the ion from the ground state to this state here, it stays in that excited state for a long time, okay? So there's a very high probability, very high chance that a second photon that comes in finds an excited ion, okay? It finds the ion not in the ground state, but in this state here. And then it pushes it upwards. And you can repeat that onwards, right? And eventually, it will emit. There's other processes that happen. It's not as simple as this, but that's the basics of it. So it's just, you're just adding photons. So you're not breaking any laws of physics. You're just adding photons.
So that's not really anything like thermal excitement happens in a reactor?
No. It's not thermal excitation. No, this is all optical, all luminescence based.
Thank you very much.
One more question here.
Fiorenzo, congratulations, amazing work.
Thank you.
When you started motivating the study of theranostics, from what I could understood, you mentioned that in biology, I guess early detection would mean the characterization of temperature gradients. I think it's something, I was really interested in that comment. If that happens, and then I understand your entire approach in the sense that you're designing nanothermometers, and then at the end it was amazing how you can actually direct them with the stem cells to where you want them, and then they can be used as nanothermometers. But then I'm trying to go back to the original comment. So I'm just curious about this. So how does it work? This idea that early problems in molecular biology give a rise to a temperature gradient in the first place that you will then assess and then solve.
So the biological reason, I don't know. I mean, speaking with my biologist colleagues, they've told me if you can design a thermal probe that can detect small changes in temperature, you're in business. Why it happens I'm not sure. Biologically, I don't remember. But I can ask and get back to you. But every illness, from what I was told, every illness has a thermal gradient at the beginning. So if we can detect these thermal gradients, when there's a doubt, when there's obviously you need of course this is in conjunction with medical doctors, you need to be able to diagnose, a potential disease. But if you can send in a nanoparticle, if the MD, the medical doctor, thinks that there is a cancer, or even before, actually we showed it in another paper. I didn't show it here, but not with these types of nanoparticles. We use a different type of nanoparticles. We can actually detect the cancer growth before conventional techniques. I didn't show it here because it's not rare earth based. We were using quantum dots at the time, but yeah.
One more question. Towards the end, you mentioned, I was also curious how you increase the ytterbium concentration, and then by way of using this multi-shell architecture, you're able to get around the problem of high ytterbium concentration and then the quenching associated with that.
So we don't get around the problem. The problem's still there, but the positives are outweighing the negatives. So when you add, so in this study here, I didn't really mention it because there was a lot of stuff to talk about, but in this study here, if I can find my cursor, there it is. In this study here, I said we made a core shell. What I didn't say, I didn't want to go into the details. This is actually an optical shell. Okay, what we wanted to do was see if we can add more ytterbium to the system to absorb more near-infrared photons. Okay, because what we saw was this was a long time ago, so we were trying to figure out the optimal doping concentration. So we were doping different concentrations of ytterbium to see if we can, ytterbium acts like an antenna. It absorbs the near-infrared photons and then transfers them to the luminescent ion, in this case is erbium. Okay? Again, I didn't talk about this, but technically what you do is you dope about 10 times more, an order of magnitude more ytterbium than whatever your luminescent ion is. For example, erbium or thulium, with the goal of surrounding that luminescent ion with ytterbium. Okay? So ytterbium absorbs the photons like an antenna and then transfers them. But the problem is at some point the intensity increases when as you add ytterbium concentration, and it plateaus, and then it dies. Okay? Because what's happening is the ytterbium is absorbing the photons, but then transferring it to each other. So it's kind of an endless loop of energy transfer, what we call self-absorption. So what we wanted to see was so this was the first time we thought of it, if we can physically segregate the ytterbium ions, we could potentially add more. So we did the standard doping at 2% erbium and 20% ytterbium, and then we added a shell of ytterbium. And we saw that it actually worked. At that time, we couldn't measure quantum yield. It was a long time ago. We didn't have the techniques, but just by the intensities, we were able to show that it worked. So with the lithium stuff, with the lithium, the ultra small particles, we did that, and we got the same effect, right? We add, you're doping ytterbium, and then all of a sudden, boom, it dies. But then, eventually, we reach 100% doping. But we can make really, really small particles. And so then we said, okay, let's try shelling it. And so we shelled. We made very thin shells, and we continuously shelled. And eventually, we had a sub-20 nanometer particle with undoped shells that were super bright. But the problem with self-absorption is still there, but just that the shelling that we did protects the ions from the environment. So the problem is still there, but just the advantages outweigh the negatives.
Alguma questão ainda? Aqui.
I work with coordination conversion in ytterbium-erbium and complexes, and one of the things I'm trying to study is the influence of the presence of fluor in the ligands of the complex.
Fluorine.
Fluorine, I'm sorry. And one of the results that I was expecting, which has already been disproven in a few of the results that I got lately, is that I thought the lesser presence of fluorine would increase the upconversion efficiency. And now you brought me this information that actually the fluorine tends to make upconversion better. I wanted to know if you could talk a little more about that, explain a little better the fluorine.
Well, I mean,
effect.
it's just a question of vibrational energies, right? So I would have guessed that the more fluorine you add in your system, I mean, I'm not a complex guy, but the more fluorine you add in your system, the better the upconversion would be.
I couldn't get a relation on that, but the ligands that I used, the energy levels, the non-fluorine version of the ligand had an energy level farther away from the conversion region. So I thought they would not. I don't know if the right word is kidnapping, but they wouldn't interfere negatively in the upconversion effect. They wouldn't sequestrate the photons.
Yeah, honestly, I don't know, because I know at some point we experimented with different ligands. So everything, if you're dealing with upconversion, every vibration you have will affect your upconversion. So I only spoke about the lattice vibration, so the phonon energy is here. But these materials are in solution. So the lattice vibrations of solvents affects quenches. The capping ligands on the surface quenches. So at one point, we experimented with using perfluorinated ligands. So just all fluorine ligands, so no carbon, no long chain carbon, just fluorine based. It was messy. It was a messy chemistry, and we abandoned it. But we did see an improvement, but just the improvement wasn't worth all the effort we had to do to work with fluorine. In complexes, honestly, I'm not sure.
Okay.
But we can discuss it after.
Okay. Thank you.
Yes. Well, thank you, Fiorenzo, for your brilliant talk and impressive results. So concerning that system that you use two wavelengths for diagnosis and therapy, and have you ever tried to combine both wavelengths and to watch some
Yes.
stimulated emission
Yes.
in the nanoparticles?
Yes, yes. So that was one of the, that was my student Artyom, that was the next step that he wanted to do, was using a single wavelength, because it's obviously it's more ideal using a single wavelength with different focal points. So for example, take a thulium ion, to get back to his question, sorry, Ayrton's question, where if we tightly focus the 980, we can get the thulium, but if we defocus, then we can get the near-infrared, potentially only exciting neodymium and not thulium. We're not there yet, but that's the next step.
Well, thank you so much,
My
Fiorenzo,
pleasure.
for
Thank you.
the talk and answer all the questions. So please join me, and thanks again, Professor Fiorenzo. Bom, obrigado pela presença de todos. Se alguém ainda quiser conversar com o professor Fiorenzo, ele vai ficar aqui o restinho da tarde, lá na minha sala, e amanhã de manhã ele está aqui também. Então, se alguém quiser conversar com ele, me avisa e a gente